Assalamualaikum and greetings to the panels. My name is Nazatul Ashelah Binti Aljahari, and today I will be presenting my research proposal entitled Antimicrobial Activity of Fungal Endophytes Isolated from Plectranthus Amboinicus. It is supervised by Dr. Muhammad Taufiq Jalil. As an introduction, let's first define antimicrobials. The Microbiology Society defines antimicrobials as an umbrella term for anything that inhibits or kills microbial cells, including antibiotics, antifungals, and antivirals. And for the last few decades, there has been a rise in a number of research conducted on bioactive compounds originating from plants and their endophytic microbes for antibacterial and antimicrobial activity purposes. In 2023, a research conducted by Sawan stated that the herb plant Plectranthus Amboinicus possesses a range of biological traits that have potential medicinal properties from its essential oil. Although antimicrobials can be obtained directly from the plant extract, this research will focus on the ability of endophytic fungi residing within the plant to serve as an alternative source because it can be easily cultured and managed. Endophytic fungi are a great source because it is capable of synthesizing many secondary metabolites, some of which can mirror the bioactive compounds found in their host plants. Moving on, there are two problems that can be identified. First is, according to the World Health Organization, the rise of additional multi-drug-resistant pathogens has become more serious as these strains are almost resistant towards most antibiotics. So that is why we need to keep up and produce more antimicrobials to combat these strains. Moreover, we cannot always depend on extracting antimicrobial compounds directly from the plant, as the yield can't always be enough. Hence, there is the possibility of us over-extracting this species, which can cause it to become extinct. Moving on, the research question involved are what are concentration of endophytic fungal extract to inhibit pathogenic microorganism growth, and how efficient is endophytic fungal crude extract as an antimicrobial agent. Next, this research serves two purposes, which is to determine the antimicrobial activity of endophytic fungal isolate, and second, to evaluate the efficacy of endophytic fungal extract as an antimicrobial agent. Next is the overview of Plectranthus amboinicus. These plants fall in the Amias order and the genus Plectranthus, but before the classification, it was named Collis amboinicus instead. Plectranthus comes from true Greek words plankton meaning spur and anthos meaning flower, referring to the spur-shaped flowers of some members of the genus, while amboinicus refers to its presence in Ambon Island in Indonesia. The characteristic of this flower is that it is a semi-succulent with velvety leaf that has soft erect hair. Its stem grows over 1 meter with leaves growing between 30 to 40 centimeters. It is most well known for its scent that smells like oregano, thyme, mint altogether. It can be commonly found around Africa, Middle Eastern, and Asia, and grows well under tropical location with warm climates and may adapt well to colder climates. According to different regions, this plant is also known as bangon, Indian borage, Mexican mint, and Cuban oregano. For the bioactivity of this plant, as many as 76 volatile and 30 non-volatile phytochemical compounds can be found. Some of them include terminaton and ergotestrol, which are known to have antimicrobial and anti-inflammatory effects. Next, endophytic fungi can be defined as organisms that colonize in healthy tissues of plants for the whole or portion of their life cycle by forming a mutually symbiotic connection with their host. Since it is symbiotic, it colonizes naturally in various plant parts without causing any illness to the host plant. A new study conducted on finding the fungi residing in Plectranthus amboinicus has found out that Penicillium chrysogenum is most prevalent, followed by Aspergillus niger and Fusarium oxyforum. The biological activity of these fungal endophytes is parallel to the plant, as they can be found to produce compounds that has similar effects. Next, for methodology, it can be divided into 6 parts. The first one is the culture, maintenance, and preparation of samples. First, the fungal endophytes isolate has been provided, so a fresh culture is needed by culturing into potato texture agar and incubating at 37 degrees Celsius for 7 days. The sample was then stored at 4 degrees Celsius until further use. They were subcultured once in a while to maintain vitality of cells. Next, for test microorganism, each species was subcultured every 2 weeks on Na for bacteria and PDA for yeast. It was then incubated at 37 degrees Celsius and at 4 degrees Celsius prior to use. Moreover, a microbial inoculum was also prepared, mixing the few colonies with 0.85% sterile physiological silane well to obtain cell suspension. Then, the turbidity was adjusted to match 0.5 McFarland standards. Moving on to the next step is the agar plaque assay. It involves inoculating Monohilton agar with test microorganism and cold boring the fungal isolates into agar plaque before placing it on the MHA. It was left overnight at 4 degrees Celsius to allow the diffusion of its bioactive substance into the medium, and then subsequently incubated at 37 degrees Celsius for 24 to 48 hours for yeast and 16 to 18 hours for bacteria. After incubation, it will form inhibition zone, which is needed to be measured to find out which endophytic fungi has the highest bioactivity. After selecting the most prominent endophyte, fermentation and extraction process is next. The selected agar plaque was placed into Eden mayo flask alongside 100 ml of yeast broth and was then incubated for 16 days at 30 degrees Celsius. This is where the fermentation happens. After incubation, the mixture was filtered to separate fungal biomass, and an amount of ethyl acetate was added to form organic suspension layer. This layer was then concentrated, collected, and dried in a fume hood to obtain ethyl acetate crude extract, which will be used in this diffusion assay. The assay starts by dissolving 100 mg crude extract into 1 ml ethyl acetate. This solution will then be injected into antibiotic test as much as 2 TU. The test is then allowed to dry. After it has dried, it was placed on a MHA plate already inoculated with test microorganism. Along with this, there is also a chloramphenicol for bacteria and ketoconazole for yeast, used as positive control, while negative control is 1% dimethyl sulfoxide, also known as DMSO. It was then incubated and inhibition zones that has formed after was measured. Moving on to the determination of minimum inhibitory concentration, minimum bactericidal and usucidal concentration, 100 joules of each produced fungal crude extract plus 100 joules of each microbial inoculum was combined into each well of microtiter plate. It was then left to incubate for 20 to 48 hours at 37 degrees Celsius. After incubation, a growth indicator was added. The MIC value will be calculated based on the lowest concentration of fungal crude extract that inhibits any visible test microbial growth, while MBC and MIC value by no presence of microbial growth on plate agar. Last but not least, the time kill study is conducted to test different concentration of crude extract. First, 19.9 ml of molehydrin broth with 3 different concentration of extract was mixed with 0.1 ml of bacteria or yeast inoculum. A growth control was also made with 1% DMSO in MHB and SDB with microbial inoculum. As drug reference, 30 UG/ml of chloramphenicol and ketoconazole was also used. After that, the culture was incubated for 48 hours at 37 degrees Celsius. It was then set at 150 rpm of agitation in a rotary orbital shaker. Next, a viable cell count was conducted by obtaining adequate sample volume of 0.1 moles every 4 hours between 0 to 48 hours. The samples were then diluted and spread out onto fresh MHA and PDA plate and incubated at 37 degrees Celsius for 24 hours. Only plates containing 30 to 300 colonies were enumerated to calculate the colony unit per milliliter. Next, three significant study that can be outlined is that first, this research provides important information on Plectranthus Amboinicus antimicrobial activity from its fungal endophytes and utilizes it for production of natural antimicrobial. Second, recognizes that Plectranthus Amboinicus is a valuable plant and needs to be protected from the possibility of it being extinct. And thirdly, demonstrates how endophytic fungi can be the best alternative when studying certain properties of its plant host, since it can mimic the properties of its host. The outcome that we can expect from this research is that discovery of antimicrobial activity possessed by the endophytic fungi is isolated, and the demonstration of how efficient of these bioactivity compounds from fungal endophytes act as an antimicrobial agent. That's all for me. Thank you for your time and thank you for listening to my presentation. Goodbye.